All right, guys. So just for today, I'm disabling the lobby. Okay, so people can directly join in. So since some of the exams are coming up, like IBHAS, as well as let's say in LGBRIMH, and even in the live batch that we're having, so we have not covered a lot of portions yet. So because major exams are just around the corner, we thought we could learn by using MCQs and also feedback from those, like explanations along with MCQs, just like how we do for PYQs, right? So we'll be doing that with biopsychology unit. Okay. We'll start by 5:10 at the max. We'll see if more people are joining in. Just a few more minutes. Okay. So I'll just set up the Google form and things like that, and then we can start. We'll see. I'll present the Google form for now. Right. So can you guys see the screen? We have Google form. I put
Yes.
email as a requirement. So yeah, we'll start with the MCQ. Okay, so there are about, I think, 100 questions. But some of them are match the following questions. Okay, so each correct answer carries 1 point. So match the following would be like number of items which is match the following. So you cannot make it like 1 mark for match the following. So I think the total is 144. Okay. Just check if you guys can use the chat. I'm not sure if everyone can use the chat. Someone who is not part of the live class, everyday class, try using the chat. Try using the chat and just let me know if you can try it.
I'm not able to use the chat option.
Okay. So then that's an issue. So how will we decide? Or we'll just go forward with answer and explanation. All right. And in between, if you guys have a doubt, you can ask, because there are about 100 questions. If you stop for each of your responses, may be bit difficult. Okay. So is that okay? So we won't be looking at the chat and see what you guys have responded for for keeping it brief and then covering most of the questions. I'll try to explain why this is the answer and then keep moving forward. I'll do it at a slow pace so that you yourself can answer it while you're doing it. Need not necessarily be on the chat. Maybe if it's on a notebook or on your own WhatsApp or a document or something, you can parallelly try answering it to understand where you stand. I thought I enabled the chat, but right now I'm not sure if going back and doing it would save time. So people who are part of the live classes, you're kind of free to use the chat. Uh, so I didn't realize this would be one of the issues here today. Anyway, so we'll start with the MCQ. So hope it's there. So yeah, what are the 3 standard measures of sleep stages? So we're starting with sleep. So let me also pull up the notes so that you can have a parallel explanation going on. So these are the notes which we have currently. So these are basic version of the notes. So we'll switch between the MCQ and the notes to explain. Right. So we're talking about 3 standard measures of sleep stages. So you will have electromyograph, electroencephalogram, graph, you could say, and electrooculogram. Oculogram for random eye movement to pick REM stages, EEG for the sleep waves, EMG for muscle relaxation, particularly in the neck. We don't use ECG. So you could find it here. So EMG, EEG, and neck EMG are 3 standard measures. So this is from Pinal. So you'll be able to find it easily. Okay. So the right answer would be this. So now, when we try to match the brain waves with the frequencies, we know the highest would be gamma. So when a person is in excess hyperarousal, then we would have beta, high beta, low beta. There are 2 types. And beta is when you are awake, wide awake, and then you're having sensory stimulation. Okay. So then would be alpha. And then we have theta when you actually start sleeping, alpha just before sleeping or restful wakefulness. Yeah. And then delta is the waves in deep sleep. So beta is around 13. Okay. Alpha is about less than 13, 12. Okay. And we have theta around 7.5 to 3.5. And delta is much less than that. Right. So gamma is not an option here. Okay. So unlike Microsoft Forms, we don't have an option where it shows immediate feedback, but this is what it is. So you guys can go check it here. So waking phase, wakeful phase, 8 to 12 hertz of alpha. Beta is 13 to 30 hertz. Beyond that, we do have a stage called gamma, which is not mentioned here for now. And then we have theta waves, which is 3.5 to 7.5, which is what we marked. And then delta waves in slow sleep. Okay. Think that is the exact one we have marked here. All right. So REM sleep is not characterized by which of the following. Uh, it does lack muscle tone so that you don't act out your dreams. Dreams are present in REM sleep, but not exclusive to REM sleep, happens more often in REM sleep. And then we have rapid eye movement. Yes. E.g. desynchrony happens in REM sleep. All right. So when you talk about REM sleep versus slow wave sleep, we have EEG desynchrony, as in irregular. You can just see it is very irregular compared to other sleep stages, 1, 2, and 3. So there is lack of muscle tone, rapid eye movement, and sympathetic arousal also is present. And then dreams are most common. It's not that slow wave sleep do not have dreams. It can also happen there. So that would be EEG synchrony. So lack of organization and planning of dreams is due to low activation of prefrontal cortex. Okay. So during REM sleep, only the limbic system gets activated a little bit. A little bit of the surrounding medial temporal lobes, but prefrontal cortex is not activated. So that is why your dream seems like very weird. Suddenly you are in a classroom and then suddenly you are in your home and back to classroom. But it makes sense in the dream, but when you think about it outside your dream, it doesn't make sense, right? So there is no proper temporal correlation, though planning or organization, it's lacking because of prefrontal cortex not being activated. Extrastriate and striate cortex are present in the occipital lobes, so they may not have nothing to do here. They do get a little bit activated because that's how you see your dreams. So yeah, moving on. The chemical that is responsible for feeling drowsy is adenosine. Okay, so this is what caffeine blocks. So when you drink caffeine, what happens is it blocks adenosine. So adenosine is actually a byproduct of daily activity. So ATP, adenosine triphosphate. So when you have a lot of uh, energy being consumed, adenosine would build up. Okay, so this kind of promotes uh, sleep promoting neurons. So we'll talk about ventrolateral preoptic neuron. So that is also sleep promoting, as well as there are arousal promoting neurons as well. So adenosine promotes sleep promoting neurons to make you fall asleep. So this is what helps in feeling drowsy and falling asleep. Okay. So more tired you feel and more drowsy you feel, it's because of adenosine. It also suppresses a lot of cognitive activity. So, but caffeine blocks adenosine and thereby making you slightly more awake. Then, degeneration of orexinergic neurons leads to what disorder in humans particularly? It's not somnambulism or somniloquy. So they are sleep disorders of slow wave sleep. Okay, so in which there is excess activation when you're supposed to be like completely inactive. So there should be lack of muscle tone and all that, but they are not properly suppressed. So when you talk about orexinergic neurons, it is related to falling asleep. So narcolepsy is the inappropriate falling asleep at different times. You feel excessively drowsy, you have sleep attacks. You may also have uh, sleep paralysis, but that is one of the symptoms. So orexinergic neurons are involved in onset of sleep or controlling sleep. So narcolepsy is related to this degeneration in humans. Okay. If you want to look at it, where do we have orexinergic neurons? So orexin is a particular, like we wouldn't say neurotransmitter, it's a neurotransmitter or it's like a chemical. So which kind of influences falling asleep, okay, or maintenance of arousal. So lack of orexins leads to falling asleep. So if there is high amount of orexin, you're going to be aroused and awake. So when this neurotransmitter, which produces orexin, is degenerated, it leads to sudden sleep attacks, which is characteristic of narcolepsy. Okay, so this is one thing you'll have to keep in mind. So next one, okay, another massive following. Massive following with sleep disorders. Difficulty breathing is apnea. So apnea is a technical term for difficulty breathing. So when it happens during sleep, it's sleep apnea. It happens because of excessive relaxation of the muscles in your diaphragm, as well as the muscles around your throat, which are supposed to keep the airway open. So those lead to collapse of the airway and leading to breathing difficulties. It can be a reason for insomnia, but not necessarily. That usually happens in deep sleeps. Then we have sleepwalking, which is somnambulism. Difficulty falling asleep after going to bed. So that's basically insomnia. Or even delayed waking. So when you wake up in the morning and you have a lot of time transitioning into proper, awake state. So that's also a typical feature of insomnia. Or if you had some mid sleep awakening and then you're not able to fall asleep, so that can also be insomnia. I mean, these are characteristics of insomnia. And sleeping at inappropriate times is narcolepsy. Okay. So any doubts or something so far? No, right? Okay. So if you guys do have doubts, feel free to just pause and then not pause. Ask a question, unmute and ask a question anytime. All right. So raphe nuclei is present in the brainstem and it is involved in serotonin production. So which is what helps uh, in a way for sleep also by controlling the amount of awakening or arousal that you have. All right. So these axons from raphe nucleus, so they project to many parts of the brain, which make you feel awake. So this is part of the arousal set. So we'll talk about that on and off switch later. So next one is slow wave sleep occurs when neurons in the dash area become active. So this is what is the ventrolateral preoptic area. So this is part of the on stage of sleep. So we have neurons which produce chemicals or neurotransmitters for arousal, right? And we have neurotransmitters which produce chemicals for or um, neurotransmitters for sleeping, which reduces arousal. Okay. The slow wave sleep particularly is related to the ventrolateral preoptic area. So this suppresses the area of uh, those arousal producing neurons. So acetylcholine, norepinephrine, histamine, even serotonin, and also orexin. So they are all like arousal systems. So the sleep promoting area in ventrolateral preoptic area suppresses them. The other way around is also true. That is why these are called flip-flop circuits. So one suppresses the other, and the other also, when it's active, suppresses it in return. Okay. So this is it about slow wave sleep or deep sleep. So ventrolateral preoptic area is involved here. Okay. So moving on, dash hormone appears to be involved in synchronizing circadian rhythms. So circadian rhythms are the 24-hour cycle, which has a lot of influence based on environment by zeitgebers like light, temperature, and so forth. So which hormone is involved? It's not norepinephrine. It's kind of a hormone as well as a neurotransmitter. Serotonin, it's not epinephrine. It's melatonin, which is particularly sensitive to light. But interestingly, even in blind people, melatonin, or not 100% blind people, people who cannot identify things or see things clearly also, because even if they're blind, they are still able to recognize the amount of light around them. They may just not see things clearly. So even in them, melatonin has an impact on synchronizing circadian rhythms. So when you move on to another time zone where you know there may be late sunrise or earlier sunrise, your initial difficulty would be in falling asleep at sun time or sundowning time or in the evening. So melatonin adjusts yourself to that circadian rhythm. So even in blind people, this adjustment is seen. Okay. So this is about sleep disorders, not disorders, sleep related MCQs. We are keeping it brief so that we can cover everything in biopsychology. Is there any particular question in here that you have any questions or doubts about? Can we move on to another topic?
I generally get confused about melatonin and melanin.
Okay. So melanin is the hormone for skin, which produces the dark pigment. It's the pigment itself, right? I understand because it's very similar. So melatonin is for light and then adjusting your sleep. How can we differentiate them? Is there any mnemonic we can do? I'm not sure. But anyway, melatonin is about that can be a possible distractor, which may lead a lot of people away into choosing melanin. Okay, so not sure what sort of a mnemonic we can have here. But anyway, someone else had a question, Adishri.
Yes, sir. So I wanted to confirm that the frequency of gamma wave rays are 30 hertz, more than 30 hertz.
Yeah, so it'll be more than 30 hertz because till 30
Oh,
is still in beta. That is high beta.
yeah. Okay, okay. Thank you,
Okay,
sir.
so gamma will be more than that. We didn't have an option here, as in I didn't make an option here. Okay. Fine. Any other questions? So we are not covering the entirety of sleep. It's not like a class. Otherwise, it'll take a long time with just sleep. All right. So we'll move on to the next topic. If you want something in depth, a particular topic, then we can definitely have a discussion. Right. So now we'll move on to genetics and behaviors. Let me just go to that PPT. All right. So the first question is about chromosomal abnormalities. All right. So particularly sex chromosome abnormalities. How do we get this one? Yeah. So it's a match the following question. So we have Klinefelter syndrome, triple X syndrome, Down syndrome, Turner's, and Superman syndrome. So we have descriptions on this side. So there are only four. So we'll only choose four of these terms. Additional X chromosome and small testes. So obviously it's a male. So it is not triple X syndrome. Down syndrome is about autosomal chromosome abnormality. Turner syndrome is also sexual, but it's most likely to be Klinefelter's, where a male is there and they have an extra X chromosome. A female with short stature, webbed neck, and sterility is Turner syndrome. So Turner syndrome is one of those syndrome where they may require hormonal replacement therapy also, or they are sterile. Okay. So in female with extra X chromosome, a female obviously means XX, another extra X is going to be triple X syndrome. Okay, so they may also have menstrual irregularities, but may not be sterile. And then lastly, we have flattened face, small head, trisomy of 21st chromosome. You would all know as uh, Down syndrome. And lastly, we do have Superman syndrome, which is basically a male with extra Y. Okay. So here also they may not be infertile, but they may have other complications like, anyway, we look at it, other complications like IQ issues, learning disability, and things like that. So this would be typically Superman syndrome. We have triple X, where they may not require much of hormonal replacement. Klinefelters may also not require hormonal replacement, but they may have issues related to secondary sexual characteristics. It is Turner syndrome and Fragile X. Oh, sorry, it's only mostly Turner syndrome, which may require them significant hormonal therapy because of sterility. All right. So Fragile X is where it could be either male or female. Most likely to be male because they just have one X chromosome, and if it is underdeveloped or uh, not properly expressed, so we can have Fragile X syndrome. Right. So these are the most likely sex chromosome abnormalities. So yeah, moving forward, a numerical value of proportion of variability that occurred in a particular trait because of a genetic variation. Okay, so basically they're saying the amount of variation attributable to genetic factors. Okay, so it's a numerical value. So that is usually heritability estimate. So the lack of variability or variability because of changes in genes. So that would be heritability estimate. Multiplier effect is basically when you have genes which enable you to express certain behaviors. You choose careers or you choose interests which align with them. So that multiplies the expression of the gene. So that is multiplier effect, which is not a numerical value at all. Inheritance quotient is just a layman's term. So it is just something I put as a distractor. Okay, so it is heritability estimate. Then find it. Yeah. Okay, so this is the famous Minnesota study. So heritability estimate is 0.7 for IQ in his study. Of course, it varies for various studies. It's a numerical estimate of the variability proportion because of genetic variation. Okay, so that is the definition of heritability estimate. So a pictorial display of a person's family relationship and medical history is genogram. It's not family tree because family tree doesn't talk about disorders and things like just people. Okay. And who are those people? Family history is a detailed history. It's not necessarily a pictorial display. Gene tree is similar to family tree, but gene tree is just a layman's term. So it's genogram. So we don't even need to go to the notes for this. And lastly, we have study of monozygotic and dizygotic twins who are separated at birth and reared apart. Okay, so this is not twin studies because they're not just comparing monozygotic and dizygotic twins. They're also separating, okay, they're not separating them at birth because that will be unethical, who may have been separated because of various reasons, particularly in the US because of uh, giving up for adoption right after birth and them going into foster families and then separating. So those things do happen commonly in the US. So it is adoption studies. So not only do they compare monozygotic and dizygotic twins, they're also comparing them when they're adopted by different families. So how do they, you know, uh, compare with each other? So linkage studies is about, how do you say it? It's about the amount of chromosomes that get linked to it and then leads to expression. That is not related to twin studies. There should be somewhere here, linkage studies. Right. I'm not sure where I've put it, but anyway, so the answer is adoption studies. Family studies basically in general is about uh, using taking a family history and using genogram to say in first generation relatives. So, or second generation or third generation relatives, this is the incidence or prevalence of this particular disorder. So you are also more likely to have this disorder. So that's about family studies. So we can expand it with twin studies. Even further, it will be expanded to capture environmental influences by adoption studies. All right. Before we go on, any doubts in this one? These are the commonly asked questions. Of course, we are not again going deeply into genetics and behavior. Any doubts or questions in this one? Nothing. Is it all clear? Any more explanation you want for a particular question?
I just have one question.
Yeah.
Do we also need to know where the melatonin is secreted?
Not necessarily. Even I'm confused. Where is it secreted? Is it hypothalamus or pituitary? Wait. Good question. So we can find out where it is before we move on to this. Current is what? Okay, so we are talking about circadian rhythm, right? So this is where it should be somewhere. Okay. So in the hypothalamus, you do find melatonin. That is what I think. So the dorsomedial nucleus of hypothalamus. And then from there, you go to the regions involved in sleep and waking. They themselves do not trigger melatonin. So it should be at the hypothalamus where you have melanopsin, which is a precursor, and then you have melatonin. Okay.
Pineal
Anything
gland is
else?
also the part of it, no?
Yeah, just checking it. But we'll resort to Google now. Right? Melatonin secretion site, right? Wow. Oh, pineal gland. Okay. So melanopsin to melatonin. So how does it convert? Opsin. The retinal ganglion cells themselves contain melanopsin. Okay. Right. So I don't know why my page is so weirded out. Okay, so we can have pineal gland as one of the major areas. So then I'll have to update the notes also. So it's not only the hypothalamus, we also have pineal gland involved in producing melatonin. Okay, good. So there's one more thing we can use as MCQ, right? Fine. So anything else? We'll move on to emotions for now. Is there any other question? So we'll
That's
open
all.
up emotions. Okay, cool. All right. So when you talk about controlled responses, emotions, and controlling those emotions, so which part of the brain is involved in controlling emotions? We have amygdala, we have preoptic area, we have prefrontal cortex. Particularly in anxiety, let's say. Okay. So the central nucleus is about producing behaviors. Okay. So not just responses in general, heart rate increase and all that. It's about actions or behavioral tendencies which are enhanced by the central nucleus of amygdala. So the lateral nucleus is about producing the responses or increasing those responses towards or increasing your sensitivity for those fear responses. So it's VMPFC which controls and inhibits your amygdala. So it's like prefrontal cortex exercising control over the amygdala, right? So this is how most anxiety disorders are kind of conceptualized. So you have hyperactivity in your limbic system, which involves amygdala, and then underactivity by your prefrontal cortex, which does not check it appropriately. So if prefrontal or ventromedial prefrontal cortex is again stimulated, you may have extension or controlled responses, even to an extent where it can lead to suppression of feared responses and some sort of a altruistic behavior like risk taking. So we'll find that here. Alright. So the central nucleus of amygdala produces fear-related behaviors. So if you have lesions there, you have less fear-related behaviors being produced. So the lateral nucleus of the amygdala producing fear conditioned emotional response. So like the sensation of fear and let's say your heart rate increase, breathing difficulty, and so many things. So it's the ventromedial prefrontal cortex which exercises control over your emotional response. So it inhibits your amygdala activity, right? So that will be the answer there. So moving on, low levels of which neurotransmitter leads to aggression? So people do say some sort of dopamine is involved, norepinephrine is involved, but there may be excess of them. But low levels of serotonin is what usually leads to aggressive behaviors. Yeah. So activity of serotonergic neurons do inhibit risk-taking behaviors, including aggression. So any destruction of the serotogenic axons leads to low levels of aggression, and then it increases risk-taking behaviors. Okay, so this is seen in monkeys also, where low CSF levels of 5-HIAA, which is a metabolite of serotonin, which gets converted later. If low levels are present in monkeys, so they were more likely to be risk-taking and aggressive. So this was one more finding. Okay, it was also found in rats and other mammals. So it is serotonin. And then we move on to blind side. Blind side is where there's cortical damage, but people can still see. All right, but if you ask them, they say, no, I cannot see. If you ask them to navigate, they'll still navigate properly, avoiding all obstacles and all that. So that's called blind sight. That is also applicable for emotions. So if you ask a person to say who has visual cortex blindness, to say what is the other person doing, what is their facial expression, is it smiling, frowning, crying, something like that, they'll still be able to say it, but they will not have conscious recollection of it or conscious awareness of looking at it, but they'll accurately say it without having this conscious awareness. So this is called affective blind sight. Okay, so emotional blind sight is just a layman's version of it. So I just put it as a distractor. So the right answer here would be affective blind sight. Okay, so it is because there is a subcortical structure which goes into your amygdala. So amygdala is what processes emotion, right? So instead of going to your occipital lobe, visual cortex, and then coming back to amygdala, so we do have direct subcortical connection. Yeah, here. So it is the pulvinar nucleus in the thalamus that directly provides this information to the amygdala. So even if a person has visual blindness, visual cortex blindness, they are still able to get this information directly from the subcortical structure. Okay, so this is what is called affective blind sight. So this would be 5, no? Okay. I'm not able to go there. Just a second, guys. All right, now I'm able to choose. So the next one, the dash system is involved in recognition of emotion. It's not a visual system per se, it's a set of cells. So you have various layers within the cortex. So some cells are uh, bigger, which are for movement, and some cells are particularly tuned for identifying shades and also discriminating between them. So it is the parvocellular, which includes colors and differentiation in shades. It provides information to the temporal lobe and to your fusiform gyrus. So it is not movement. Unicellular is a combination of both. So it's a parvocellular part of the visual system, which is involved in recognition of emotions rather than magnocellular. Magnocellular is for movement. All right. So then people with DASH can move their facial muscles voluntarily, but not in response to an emotion. Right. So this is there are two types of facial paresis. So it's not paresthesia. Paresthesia is basically pins and needle sensation, that tingling sensation when you know have lack of blood flow in the area. So that is not what it is. It's not paresthesia. It is because of paresis. Paresis means weakness of muscles. We're talking about emotions, right? Yeah, here. Right. So there are two types of paresis. Sorry, emotional and volitional paresis. All right. So in emotional paresis, they may not be able to express an emotion, but they may move their facial muscles voluntarily otherwise. Okay. But on the other hand, when you ask people to move their facial muscles voluntarily, people with volitional facial paresis may not be able to do so. But when they have an emotion, they may automatically smile. They may make facial expressions. But if you ask them to do it voluntarily, they may not be able to do so. So that's the opposite. So the question is, people with DASH can move their facial muscles voluntarily. So it is not volitional, but not in response to emotion. So it will be emotional facial paresis. Okay. So definitely not paresthesias. Right. We have two more questions here in this particular topic. Left half of people's faces are more expressive than the right. And reason being, the right hemisphere is involved in emotional expression more than the left hemisphere. So which of the following are true? Is it only assertion, only reason? Both are true or both are false? Right. So this is kind of like in the pattern of net because net has more analysis based questions. So here the left half of people's faces are actually more expressive. The reason that we have more right-sided structures which are involved in emotions. For example, even in speech, the intonation and expression and all are taken care of by the right side of Broca's area. So there's this theory that left side is more for sequential, detail-oriented things, and right side of the brain is more about parallel processing and about processing the whole. Okay. So emotions come into this domain of a wholeness, right? So we'll see here also, not this. All right. So the left half of people's faces and even other primates are more expressive. So you'll try to find that people are able to understand and express emotions more with their right side. All right. So that is one of the reasons. So both A as well as R, assertion and reason, are correct, and it is the explanation also. Right. So last question. Dash of the amygdala is involved in fear-related behaviors or producing fear-related behaviors, and dash of amygdala is involved in producing conditioned emotional response. So we already saw this. Central nucleus of amygdala is involved in fear-related behaviors, and lateral nucleus is about conditioned responses. So it can be emotional arousal and those things, not the actions. So we are talking about physiological responses. So this will be the correct answer. So we can verify it also. Right. Emotions, right? Why we are having sleep. Yeah. So central nucleus of amygdala is about producing fear-related behaviors. Lateral nucleus is about producing conditioned fear-related emotional responses. Okay. All right. So any doubts in any of these questions or any particular topic you want more details on before we go on to the next one? Is it clear? Do you guys need any more explanations or anything? Can we move on to the next topic? Is it clear?
Yes, sir.
Right. If more people said so, it'll be easier and faster. All right. Even you can just put a thumbs up. That's also okay. Anyway, so we'll go on to sensation. This is about sensory systems. There are quite a few PPTs that are there. So we have visual system, we have auditory system, and even chemical senses. So we open them and keep them on separate tabs. Okay. So match the sensory pathway with the correct thalamic nucleus. So we know that thalamus is the relay center of the brain, right? So whatever senses you have goes through the thalamus and then to various cortical areas. You do have some subcortical connections directly from thalamus. For example, the pulvinar nucleus, no? So it sends information to amygdala. Pulvinar nucleus is a part of thalamus. So we do have other projections rather than the cortical structures also. So anyway, the visual cortex is basically lateral geniculate nucleus, and medial is auditory, and medial dorsal nucleus is olfaction, yeah, here, and VPN, ventral posterior nucleus, is gustatory. Dorsal anterior and ventrolateral is just added there, but these are the common nuclei and their corresponding sensory pathways. LGN is visual. You'll find it here. It's chemical vision. Okay. So the lateral geniculate nucleus is involved in visual processing. Okay, so that is a part of the thalamus which conducts LG. So here we have the magnocellular and the parvocellular also. So it's about the cortical structures and also in the thalamus, which has a columnar structure. Okay, so magnocellular is for more movement, parvocellular is for more color and shade differences and things. So when you talk about auditory, so I've gone to cortex already. So we have the medial geniculate nucleus for auditory. All right. So medial geniculus MGN, you could say, is for auditory stimulus. And then when you talk about chemical senses, which includes both smell as well as taste. So we have the VPN and MPN, no? Right. So for gustatory, it goes through the ventral posterior nucleus in the thalamus. And what about olfactory? For olfactory, it is medial dorsal nucleus. Okay, MDN. All right. So these are the various structures in the nuclei which connect to various different cortical structures. Okay. So olfactory is the only structure or only sensation which does not go through, for example, what? It does not directly go through thalamus. For example, it can go through piriform cortex directly, and then it goes through the thalamus. So piriform cortex is your primary auditory structure, olfactory structure. All right. So then it goes to the thalamus and then back to your orbitofrontal cortex. So if you were to say olfactory, you have kind of the two structures which are involved in the sensation, piriform as well as orbitofrontal. And it is one of the only structures or only pathways which does not go through thalamus first. That's also a possible question, which I think we do have later. Fine. So next is match the sensory system with the correct primary cortical area. So for olfactory, we just saw it, piriform cortex. Somatosensory is your post-central gyrus. Okay, so pre-central is for motor, post-central is somatosensory. Vision is your let's say striate cortex. It can, it should come here, no? So olfactory is piriform. Okay, so vision is located in the occipital lobe. So we call it striate cortex. The association is called extrastriate cortex. For gustatory, it's insular, and there are other surrounding structures also. Okay, so these are the various cortical areas and their respective sensory, modalities. So now we move on to the sense of somatosensation or touch. which of the following mechanoreceptors are involved in pain and temperature? For pain and temperature, it is free nerve endings. Ruffini corpuscles can be about, what is the exact word? Okay. So Meissner's and Merkel's disc are about stereognosis, about the pressure, edges, and form of things. So Ruffini is about, what is the exact word? It's a much deeper structure. I'm not able to find the exact word. Let me just pull this up. We have it in another place. It's kind of at the tip of the tongue. I'm just not able to get that. Auditory and somatosensory. Okay, it should be here only. So Ruffini corpuscles, I know they are deeper structure, but what do they involve themselves in? All right, so we have Ruffini corpuscles here. So they're about skin stretching and also proprioception. So Ruffini endings are slightly more deeper than your Merkel's discs. So they are in static forces when your skin is stretching and all that. Okay, cool. So moving on. Which of the following mechanoreceptors is involved in stereognosis? Okay, so stereognosis is basically when you have an object in your palm and identifying what object it is. It involves identifying structure or surface and also its form. So Meissner's and Merkel's discs are involved there, not free nerve endings, not Pacinian, or even not Ruffini. So if there are Merkel's disc also, that would also be involved, which is not there. So it's only Meissner's corpuscles, right? So in the visual system, the dash pathway is what pathway and dash pathway is where pathway. So what pathway helps you identify what the object is. When you see something, it helps you identify what the object is. The where pathway is when you're able to identify how far away an object is, in which direction the object is. So the where pathway goes to your temporal lobe where you have verbal information, memory for those things. Okay, so it is more ventral on the underside. And the parietal lobe is useful in identifying where an object is located. So it has to go upward, right? So that will be the dorsal side. So ventral pathway is where, sorry, what pathway? And the dorsal, the top, is where pathway. Okay, so we can find this, closed it. Just to give a picture so that you can like encode it better. So from the cortex to association areas, so here you have it, the dorsal and ventral streams. So the dorsal is above. So if you know the directions within the nervous system, dorsal is top, ventral is bottom. All right. So the dorsal stream goes to your parietal cortex, parietal lobe, which involves in identifying where an object is. So visual spatial pattern perception, and the ventral goes to your temporal lobes to identify what it is. So that is called the what pathway, right? So this is the ventral stream. So now the smallest body in the bone is the three bones that are in the ear. But even within them, the one at the end, so malleus, incus, stapes. Okay. So stapes is the smallest bone. So there are three bones which conduct from the ear membrane to your oval window, which connects to the cochlea, right? So stapes is the last part of it, which is the smallest bone. So if you want a picture of it. Oh, why are you still in vision? Oh my God. Audit. Right. So here you have the tympanic membrane, and then we have hammer, anvil, and stapes. So hammer is malleus, anvil is the incus, and stapes is the stirrup. So which is the smallest bone in our body. Okay. So together they are all small bones, but the smallest is stapes. Right. And moving on, match the sensory system with the respective organization principles. Okay. So you know that the retina and the visual cortex are kind of mapped 1-to-1. So it's called tropism. Yeah, tropism. Right. So that is called retinotopic. So that is in the visual field. And in audition, it is about the frequency. So lower frequencies are on one side of the cochlea, and higher frequencies are on the other side of the cochlea. So they are tonotopic based on frequency. And somatosensation is somatotopic. As in, you can see the, what do we usually call it? The homunculus. Okay. The homunculus is literally a mapping of your body on the somatosensory cortex. It's there for the motor cortex also. So if you look at it, you'll be able to identify it. So auditory is tonotopically arranged based on frequency, and somatosensory is somatotopically arranged, where you have the homunculus, where the head is on top, below is your neck, and other things. So you'll be able to find it in the cortical structure. Yeah. So this is an example of the homunculus. Of course, it's not a smooth, map of your body. So of course, there is some differences where, head is way beyond your hand and all that. Okay. So particularly in the homunculus, some parts of your body like lips and hands, they have more sensations. So they have more areas in the somatic cortex, somatosensory cortex, right? So for audition, okay. So auditory cortex is tonotopically organized. So it depends on frequency, on the basis of frequency. So even the basilia membrane or the hair cells that would be here, they're arranged in a way that shorter cells are earlier and the longer hair cells, they're way more at the end so that they can vibrate and pick up different frequencies. So that is what is the arrangement that you also see in your auditory cortex. Okay. For vision, it is retinotopic. So it's almost like a one-to-one mapping. And where do we find that? See. So whatever you see here is exactly what it goes to the other side. So the same mapping on the retina kind of gets mapped 1-to-1 in your striate cortex. Okay, so that is retinotopic organization. We don't have such an organization for smell, but there seems to be some sort of a relationship between a species having very similar organized structure versus some other smell sort of a thing. So between smells, we don't have any differentiation. But within a species, there seems to be some sort of a similarity in organization, but it doesn't follow any principle as per se. So gustation also doesn't have any particular organizing principles in their uh, cortical systems. Right. So moving on, the order of cellular cells in the retina. So I remember it by the acronym GABH. So ganglion, and then amacrine cells, bipolar, and then horizontal cells. So you'll be able to find it in the retina's picture. So you have the back of the eye here. So first layer is ganglion cells, bipolar, and then horizontal cells. And lastly, you have the receptors. And then you have the back of the eye. So the ganglion cells kind of collect all these information because these are all transparent. So through these light passes and then hits the photoreceptors, rods and cone. And from them, these get electrical signals. And all of them are collected at the ganglion cell. And then they go through a particular point, which would be your blind spot. Okay, so this is the blind spot. They all collect and go through the blind spot. All right. So GABH is one acronym that I used to use for remembering this. These are all like most commonly asked in NET, maybe sometimes in MPhil also. So which is the only sensory system that does not pass through the thalamus before reaching its cortical structures? So that will be olfactory. It goes to piriform cortex and then through thalamus and it goes to orbitofrontal cortex. So olfactory is the exception here. Everything else goes to the thalamus. Okay. Auditory system goes through medial geniculate nucleus, and visual system goes to lateral geniculate nucleus, and gustatory system goes to VPN, ventro posterior nucleus. Okay. So which of the following is responsible in sound localization by mapping an auditory space? So that would be your superior colliculus. Superior olives are also involved, but from them you or your brain sends it to superior colliculus so that you can map it and then you're able to identify in a three-dimensional space. Sound localization. Where did I put that? Huh. So initially it is mediated by superior olives. So when a sound originates in the left and right ear, there are differences, right? So it'll be louder on the ear that is facing towards the source of the sound and less louder on the other side. Similarly with the amount of pitch and integrity of the sound quality. So from then on, this information goes on to your superior olives. So they kind of make an auditory map around you. So then you're able to identify where it is exactly coming from. It also goes to the inferior colliculus, but it is your superior colliculus that helps in locating this through a auditory space or a map of auditory space. The answer would be superior colliculus here, right? So how many do we have? We still have a few. After this, we may take a small break, okay? So tendency of acoustic stimulus, basically sound-based stimulus of different frequencies to cause different parts of the basilar membrane to flex back and forth is called place coding. So, so higher levels of frequencies or higher frequencies would be identified at a different location. Acoustic stimulus of different frequencies causing different parts. So that is basically place coding. All right. So higher frequency sounds, they are identified and picked up by hair cells at the particular location of basilar membrane, and low frequency sounds as the end of the basilar membrane because of the differences in hair size. They are more sensitive to picking up either higher or lower frequency of sounds, and they are placed in a particular order within a particular location of the basilar membrane. So this would be place coding, right? So then we have dashed layer is sensitive to color, which is parvocellular. Dashed layer is sensitive to movement. It's magnocellular. You can understand by magno M is to movement M. So that is one way of saying it. So coniocellular combines both of them to an extent. So dash is rich in dash, and is responsible for color and detailed vision. Okay. So cones are the ones that are usually responsible. Okay, no. Rods or cones. Right. So fovea is the one where you have the highest amount of cones. Also rods. Okay. So rods are about sensitivity to light and intensity of light. So you have cone cells, which may be in different for colors. Different colors have different cone cells. So it's fovea and cone cells. Hope I'm right. Yeah. So cones are also responsible for color vision. Right. So there's another way to call it scotopic and phototopic vision. Okay. So scotopic would be your night vision, intensity of light. Phototopic is for color vision. So that is also one common term that used to be coming in net. So fovea is where you have the intense collections of rods and cones. Right. It should be somewhere around here, fovea. And cones particularly are more concentrated in your fovea. Okay, moving on. So correct order to the auditory receptor, which is basically the basilar membrane inside the organ of Corti. That is the last part. And it starts with pinna, your outer ear. So how do we go from pinna to your organ of Corti? So from pinna, we go to tympanic membrane. From there, you have the three bones, which are ossicles, and then it goes into the oval window, and then goes into the snail-like structure, which is cochlea, within which you have organ of Corti, which includes the basilar membrane, the hair cells, and so many things. So this would be the correct one. I'm not sure if I kind of turn on the option for shuffling the options, But anyway, it's the first option. So just like the ventral and dorsal pathway, we also have anterior and posterior pathway for the auditory cortex. So the dash stream of auditory pathway is the what pathway, and the dash stream is the where pathway. So we know the primary auditory cortex is in the temporal lobe. So from there it goes to the temporal pole as well as your prefrontal cortex. So where it is analyzed to identify what it is. So it goes more forward, and then from the Hessel's gyrus, which is basically your auditory primary auditory cortex, it goes to your parietal lobes. So it is kind of like rear. So it's posterior, and it helps in identifying where sounds are coming from, the source of sounds. So that is the posterior pathway. You can find an image also here. Right. You have your anterior stream, which goes from the Heschel's gyrus to more anterior sides of the temporal lobe, and also some part of your frontal cortex. And then from again your Heschel's gyrus, it goes rear to your parietal lobe, which kind of helps in identifying location. Okay, so that is your where pathway in a way. So how many do we have more? We have 2 more. All right, good. So the anterolateral system in somatosensory pathways carries pain-related information, so nociception. And there's a dorsoventral or dorsolateral, I think. So that also carries other touch-related sensations. So vision, then we'll change it to somatosensory. Just a second. So there are 2 parallel pathways from the skin and the basically your spinal cord to your cortex, basically somatosensory cortex. Okay. So you have the anterolateral system, and you also have the dorsal column medial lemniscus system. Okay, that's a mouthful. Anyway, so the anterolateral system carries temperature and pain-related stimulus or sensations. Then we have the dorsal column medial lemniscus system. It carries about touch and proprioception, nothing intense and things like that. So this carries from the spinal cord to your hindbrain and to your somatosensory cortex. It's more anterior and lateral. So your dorsal, which is more on the back side, this carries to the medial and lemniscus system. So you have medial lemniscus, which collects various information. So you have 2 parallel pathways for somatosensory information. One is for just touch and proprioception, and one is for pain and temperature, so that you can react quicker. Right. So, and lastly, we have this master following. You guys can also pitch in for this. The inability to smell is about not smelling things. So it usually happens when there is a whiplash sort of an effect, and is called anosmia, because there's a plate called cribriform plate from which or through which you get information that goes to your the chemical senses, no? Information that goes to your piriform cortex. So those axons get shattered or they get stretched. So that is why people will not be able to smell right after any sort of a concussion or a small accident. Yeah, so this one. So you have the olfactory receptor and you have the cribriform plate through which it goes. If there is any whiplash, a concussion, or even any head injury that happens, coup and counter coup may happen, but that is at the cortical level. So we have this plate which can move backward and forward. There can be shearing of these axons, which leads to anosmia. So anosmia is basically inability to smell, and inability to taste is called agwasia. So that will be anosmia, inability to taste, agwasia, inability to recognize objects by touch. We already saw this. It's stereognosis. So it will be astereognosia. Okay. Failure to recognize parts of one's own body is asomatognosia, and inability to recognize faces is prosopagnosia. It is related to the fusiform gyrus, especially on the right side. It's kind of under the temporal lobe. Okay. Anosognosia is inability to recognize your own symptoms. That could be for any modality or any set of disorders. So we may not have a particular region for that. Okay. So this is it about sensations. Or any doubts, any questions, any particular topic you want me to explain further? Is it all clear so far? Are you guys able to understand? Anything, guys, before we take a break and come back? Is it all clear? All right. So what we'll do is we'll take a brief break. It's already 6:11. Maybe we'll come back by 6:50. Okay. Or we'll take a 9-minute break. We'll come back by 6:20. I just need to drink some more water and all that. So we'll come back by 6:20. Okay. 8 minutes. So we'll be back by 6:20 and continue. In the meantime, if you have any doubts or questions, do put it in the chat or keep a note of it. When we come back, we'll discuss that. All right, guys. So hope you're back from the break, and we do have a question in the chat. Guys, if you have any other questions, you can directly drop me on WhatsApp because the chat is not working here. Right, so we'll address this question. So this A, how do we remember A-based terms? All right. So basically A means lack, or it could also mean acquired. Right. So I'll try to bring one more slightly tangential example here. So dyslexia versus alexia. So dyslexia is about having difficulty. Alexia is something acquired because of some brain damage or something like that. Okay, so dyslexia is kind of innate. So A basically means a lack, but kind of acquired, absence of it. So for smell, it is like straightforward: nose, nosmia, smell. Okay, anosmia. And somatognosia. Agnosia basically means identifying. So if we have prosopagnosia, that means agnosia for faces. Pros is kind of like for faces. So that is how you'll have to remember. Basically, agnosia means difficulty identifying something or difficulty recognizing it. All right. So when you say stereognosia, we already knew stereognosis basically is for touch. So astereognosia is going to be absence of recognition by touch. Anosmia is smell, we already discussed. Aguasia is for taste. That requires you to understand, what do you call it? A little bit of Greek, maybe. Maybe gustatory is G, aguasia. So that could be one way to remember it. Okay. So this is how I remember it. So yeah, of course, it takes repeated practice. Another way is to keep checking yourself with tests. You don't have to reread. So there are like a lot of literature which says reading does not help you. Reading once, testing. Again, going back to read to understand and then testing yourself is way better. So keep testing yourself using such MCQs and things like that. Okay. So that would be one of my suggestions. So any other doubts or questions, guys? Is it clear? So can we move forward to the next topic? Any other questions? You can unmute and ask your questions also. Fine. So we'll move forward. We didn't. Oh, so the dash stream of auditory pathways, what anterior, where is posterior? Nothing else we have missed out, right? Okay. So now we go on to neurons, the structure of neurons and how do they conduct, action potential, and how do they carry and all that. So part of the nervous system responsible for conservation of energy, basically by relaxation, is parasympathetic. And those for allocation of resources to expend energy is sympathetic. We already know this, so it's pretty simple. We're not going to get information from the slides for this. Now, which neurotransmitter undergoes recycling by enzymatic degradation exclusively? So it will be acetylcholine. Acetylcholine esterase, and there are various enzymes involved in degrading acetylcholine. It is taken back and then reconfigured in the neuron. So that is acetylcholine. So then we have DASH is predominantly an excitatory neurotransmitter and DASH is an inhibitory neurotransmitter. So I remember in the first mentoring session, you guys kind of confused me also when I was clear. So GABA is the inhibitory neurotransmitter because it is implicated in a lot of anxiety where you have a lot of thoughts and arousal and your ventromedial prefrontal cortex is not able to inhibit it. Okay, similarly in OCD also. But for excitation, it is glutamate. Right. So glutamate is the major excitatory, predominantly excitatory. GABA is predominantly inhibitory. So which of the following ions is involved in exocytosis? Exocytosis is basically where a neuron releases neurotransmitters into the synapse. So that is mediated by calcium ions, not sodium. Sodium is implicated in the action potential. Potassium and chlorine are both implicated in reaching up to the action potential and also in action potential too. But sodium is mostly about conduction of action potential. So this is about exocytosis. So it is calcium. Which of the following are not catecholamines? So there are two kinds of amines, amine-based neurotransmitters. You have indolamines and catecholamines. So indolamines include uh, dopamine, norepinephrine, and epinephrine. Catecholamine includes, sorry, catecholamine includes epinephrine, dopamine, and norepinephrine. Indolamine includes serotonin. Okay. So which is not a catecholamine here, GABA. So GABA is a likely a monoamine neurotransmitter. We'll check that later. Okay, so catecholamines are these. Indolamines are serotonin. So the neuron, when it is at rest, has a resting membrane potential of minus 70 millivolts. It is minus 70. It is negative. So please don't click 70. Okay, so 90 would be a hyperpolarized, 50 would be a depolarized neuron. So 70 is also very positive. So it's kind of 50 and 70 would be very highly reverse polarized, not depolarized. And so voltage channels are dash opened and sodium potassium pump actions or functions by passive or active way. So anyway, voltage channels are channels on the surface of the neuron, which do open in a graded fashion. The amount of excitation it gets, it opens more. So it is more passive. All right. And sodium potassium pump actively consumes energy to pump out sodium and bring back potassium. So it is more active. So passive active is the answer here. All right. Then we have which of the following is amino acid from which indolamines are derived. Indolamines are basically just one. We have serotonin. Serotonin is derived from tryptophan. Dopamine is derived from tyrosine. Okay. So glutamine and glycine are monoamines. Excitatory and inhibitory postsynaptic potentials, basically EPSP and IPSP, they are decremental in nature. They are also passive and they're not active. They're not incremental in nature. So they're decremental in the sense that if they start from point A to point B, they'll lose strength. The intensity kind of gets down unless they are reactivated by another ion channel. So it is passive and decremental, not decremental only, not passive only. Right? Which of the following follows all or none law? It is not resting membrane potential because it's always there. It doesn't follow either, and it can be decreased and increased also with excitation or inhibition. It is action potential that follows all or none law. So it either gets activated completely or it doesn't get activated at all. It is not graded. So in that way, it is all or none. Excitatory and inhibitory are both graded and they are passive too. So they are not following the all or none laws. So they are more graded potentials. Next is a neurotransmitter is a or an dash of its receptor. Neurotransmitter is not a subtype of its receptor. That doesn't make any sense. It's not an enzyme. It's not an ion. It's a ligand. Ligand is basically this sort of a chemical that gets attached to only this or has an affinity for this. So in a neurotransmitter, you have receptors. So only certain types of receptors can go into this. Other types of receptors will not get attached here. So it's a ligand. A neurotransmitter is a ligand for its receptor. Okay. So then we have ionotropic receptors and metabotropic receptors. So ionotropic receptors bind to small neurotransmitters only, and metabotropic receptors can bind to large neurotransmitters also. So that is one of the major differences. Okay, so they are bigger and they can have large neurotransmitters attached to them, more protein-based neurotransmitters like peptides. So they can attach onto metabotropic receptors. So small neurotransmitters, they get attached to only smaller ionotropic receptors. Okay, so that is one major difference. So then we have varicosities. For this, I may have to show you. So varicosities are basically where, what do you call it? Neurotransmitters are released from, but they are like more elongated like a root which has a fibrous root. Fibrous is a tap root, right? So in plants, so this will be more fibrous. It spreads out more. Have that here. Okay, I am not sure if we have that here or is it neurotransmitters? I think it should be here. Hey, it's not here. I'm not sure where I put it. Anyway, so varicosities are very long fibrous sort of extensions from your axon. So they have a lot of beads on a thread sort of a structure, right? So they release neurotransmitters into the brain's fluid, not into a particular synapse. So they don't have synapse particularly. They just release them into the bloodstream and the cerebrospinal fluid within the brain and all that, right? So they are non-directed and they have monoamines mostly. So amino acids are most likely to not be a neurotransmitter. But anyway, autoreceptors are receptors in the cell which is giving out the neurotransmitter. So it is presynaptic. Autoreceptors are present in the presynaptic neuron. They kind of regulate further release of neurotransmitters. So they inhibit release of further neurotransmitters. So let's say an autoreceptor releases, let's say, dopamine. So excess dopamine goes and attaches to the, sorry, not autoreceptors, a presynaptic neuron releases dopamine, let's say. It goes and attaches to the autoreceptor because it's excess. And then now the autoreceptor gives the message that dopamine is excess. So you have to slow down the release. So that way it conserves dopamine or any other neurotransmitter for that matter. So it is presynaptic and it inhibits further release of neurotransmitters. Okay. Axonal conduction in the natural direction is orthodromic. So axon can go from, conduction of neuro action potential can go from the axon hillock to the terminal buttons. So that is the natural direction. It can also have an opposite direction. So that is called antidromic. All right. Andromic is just some distractor to confuse you. The period during which it is possible to fire the neuron. Again, this is after an action potential, but only by applying higher than normal levels of stimulation. So this is called relative refractory period. So when there's an action potential which is fired, there's a lot of chemical changes in the neuron, right? So what happens is the neuron cannot fire another like action potential immediately. But if you give it higher than the usual stimulation, higher than usual amount of excitatory neurotransmitters or excitatory postsynaptic potentials are there. So what happens is it can kind of add up and lead to another action potential even before it completely resets. That is relative refractory period. There may be a point until which there may be no possible further action potentials. That is absolute refractory period. So what we are dealing with here is a relative refractory period. Okay, it's not inhibitory period. So that's not the term here. That's not the technical term at least. Fine. So then we have about K plus ions. For this we need a picture of this. So in the neuron we have various ions. Okay, we have potassium, we have sodium, even chlorine, and even positive protein ions. So they are in various concentrations within the neuron and outside the neuron. So K plus R or potassium plus ions are mostly within the neurons, and outside the neurons, they are kind of lesser in concentration. All right. So they do not readily move out because of concentration gradient. The reason being inside of the cell is way more negative. So it would be an electrostatic force which holds particularly the protein and more amounts of sodium outside. So that leads to an electrostatic pressure. So which kind of keeps the K plus ion attached inside. Okay. So this would be K plus ions more on the inside, sodium is more on the outside. and protein which do not cross over, and they are the reason K plus are kind of held also. So this is at rest. So it would be electrostatic pressure, not sodium potassium pump, which activates after an action potential. It's not involved. Equilibrium and concentration gradient are kind of the same thing. So they are not involved or predominantly involved. In fact, they would actually push the K plus outside. They do not resist it. They do not stop them from moving outside. All right. So IPSP, inhibitory postsynaptic potentials, they make it even more negative. So that is, they hyperpolarize the cell and they decrease the chances of the cell firing an action potential. Okay. So IPSPs are inhibitory postsynaptic potentials that start from the dendrite and move towards the axon hillock. And they make the cell even more negative from minus 70 millivolts, which is the resting membrane potential, can go to 90 and even further. This makes the cell less likely to set off an action potential. So it's decreasing. Which of the following neurotransmitters are transmitted by microtubules? Okay. So neurotransmitters are basically largely two types. One is large and small. So small neurotransmitters are packed into vesicles, and those vesicles transport to the terminal buttons. But in the case of large neurotransmitters like peptides, so they are directly transmitted from the cell body to the, let's say terminal buttons, and then they are packed. So neuropeptides are the only neurotransmitters that are unpacked and transmitted through microtubules. All right. So which of the following ion is responsible for generating action potential and its conduction? All of them are kind of involved, but conduction is mostly because of sodium. So sodium enters rapidly and then it moves and then triggers further opening of sodium channels. And then again, sodium rushes in. Okay. So of course, potassium also rushes out, but that doesn't trigger further sodium channel opening. So it is mostly sodium that is involved. Right. So now we have sodium potassium pump. So it pumps out the excess sodium that has come inside and it takes back the potassium that has gone outside. All right. So it has to take in or pump out 3 sodium and then it has to take in 2 potassium to make the cell more negative. So now the cell was negative in the first place because of action potential. A lot of sodium has come inside and potassium has gone outside. Now if it equally exchanges, let's say 3 sodium for 3 potassium, then it will only maintain it. So it has to push out more sodium and take in less potassium so that the positive charge net is way lesser. And then there's a overall net negative charge. So it takes out 3 sodium and takes in 2 potassium. All right, hope that clarifies things. So myelin sheath is predominantly made up of fat. It is also made up of proteins, kind of a 80-20 divide. But it's predominantly fat and it increases the speed of transmission. Myelin sheath does not decrease the speed. It actually multiplies it faster. So the reason being saltatory conduction happens because of myelin sheath. This is also a possible question. Just a second. So you can see the myelin sheath, it basically covers most of the surface of the axon. So what happens is the sodium channels are only present at these nodes, which are called the nodes of Ranvier. So they kind of explode out, and as in like potassium explodes out, as in goes out in bulk, and sodium rushes out in bulk. And from there to the next node, they transfer like very quickly, but like EPSPs or IPSPs. They are quick, but they are degrading in strength, but they're very quick, almost instantaneous. If they are to open further and further and further channels, like here, it takes a lot of energy and time. So myelin sheath basically increases conduction by that. So you can see here, myelin sheath composes of 80% fat and only 20% proteins, predominantly fat, right? So that's what it is. So during repolarization phase, so this is after peaking of action potential, by the time it peaks, potassium has already moved out. Okay. To some extent it has moved out, but there is still potassium, and sodium rushes in. This is what leads to the let's say the action potential happening. And during the repolarization, it becomes even more negative by all the potassium moving out. So this is a part of the repolarization phase. And then the sodium potassium pump sets in, actively exchanging potassium for sodium. All right. So during the repolarization phase, at least during the peak of action potential, it is slightly positive. And then during repolarization, even more potassium moves out, making the cell more negative. Okay. So IPSPs open up. Okay, these are like postsynaptic potentials, not action potentials. Just before an action potential, what happens is more and more potassium goes out, and chlorine also comes inside. So they open up chlorine and potassium channels. Which of the following ions do not move out of the cell? So we already saw this. Protein ions do not move out of the cell at all. So chlorine does come in, potassium does move out, sodium does move in and out, either through passively or through the potassium pump, sodium potassium pump. But protein ions always stay inside the neuron. So that is the answer there. Which of the following kind of synapses mediate presynaptic facilitation? So there are various types of synapses. We have that here. Synapse, we do have it. Okay, so we have axoaxonic, we have axodendritic. Okay, we have dendrodendritic also. So we have axodendritic, which kind of attaches to the dendrite. So you can see that in this picture on the right. So the terminal buttons attach on the dendrites or the cell's body. So this is axosomatic. In axonic, what happens is that the terminal button attaches or synapses on another axon, thereby preventing it from synapsing or exocytosis further or increasing it. So you can adjust the presynaptic neuron synapse functions based on axonic synapse. So what was the question here? Which of the following synapses mediate presynaptic facilitation and inhibition? That will be axonic. So another synapse happens on the axon itself. It mediates whether that cell's axon fires or produces more neurotransmitters or lesser neurotransmitters. Okay, so that is the answer here. Then we have neurotransmitters are packed in synaptic vesicles. So it's very easy. Conduction of action potential in interneurons. So interneurons are basically neurons without axons. So they are only nuclei. They only have dendrites. So if they do not have axons, how will they conduct an action potential? They cannot. Right? So they only have graded IPSPs and EPSPs. So they have graded potentials and they are decremental in nature. So they do not have all or none action potentials at all. So they're not non-decremental because IPSPs are decremental. So this would be the right answer here. Right. So now we have Schwann cells, which is a glial cell. So it is present outside or in the peripheral nervous system, and it covers the nervous system with myelin sheath. Okay. So, but it happens in the peripheral nervous system, not in the brain, not in the spinal cord. So similarly, in the central nervous system, we have oligodendrocytes. Okay. So oligodendrocyte is in the shape of kind of like a star. Okay, we do have it here, I hope. So they're basically like 4 major glial cells. All right. So this is an oligodendrocyte. So you have it on the right. It connects several neurons at the same time and myelinates them. This happens in the central nervous system. And in the peripheral nervous system, it is more like one myelin sheath is made by one. Sorry about that. So one myelin sheath is made by one Schwann cell. All right. So for this particular reason, if there's any damage in peripheral versus central nervous system, the myelin sheath in the peripheral nervous system easily connects to the next part of it. But in this case, it has to collaborate with several other neurons. So it is interneuronal connection and conduction. It is very difficult to re-establish the connections. All right. But it is rather easier in the peripheral nervous system. So remyelination or even reattachment of a neuron to another neuron. So it's rather easier in the peripheral nervous system. So that is one major reason. And we have other glial cells like astrocytes. So the astrocytes kind of restrict the amount of blood that goes into one place or one part of the body also. Okay. They also clean up other debris. So they kind of like eat up waste bodies in the brain and blood also. So that's the next question. No, no. All right. So DASH transports the synaptic vesicles to the terminal buttons. So it is the tubules from the cell body to the terminal buttons that transport synaptic vesicles, which have neurotransmitters packed inside them. Right. So next, DASH performs the function of controlling amount of blood. We just saw it. It's astrocytes. Right. So they are supplied to various parts of the brain. Which of the following is false about interneurons? We just saw about interneurons. So they are basically neurons without axons in the brain. So they integrate neural activity. Yes, they do that. They lack dendrites. Yep. No, they do not lack dendrites. They lack axons. They are multipolar neurons. Yes, they have several dendrites. So they are multipolar in a way. They can have short axons or even no axons. Yeah, just but they do have dendrites. Otherwise, they will not even be able to connect with each other. So this is false about interneurons. All right. So lastly, group of cells in the PNS, peripheral nerve system, is called, or group of cell bodies. So there are axons and cell bodies, right? So we have axons and cell bodies, which are called different names in the central nervous system and the peripheral nervous system. If they are just cell bodies in the peripheral nervous system, they are called ganglia, which was the question. In the central nervous system, they are called nuclei. For example, the lateral geniculate nuclei, they are just predominantly just cells. And in the central nervous system, axons are called tracts. So white matter is basically tracts. And in the peripheral nervous system, they are called nerves. Okay, so that is one major distinction here. So group of cell bodies in the PNS would be ganglia. So one misnomer here is basal ganglia, which is predominantly cells, but within the central nervous system, but it is still called basal ganglia. So that is a misnomer. So doesn't mean basal ganglia is in the peripheral nervous system, it's still in the central nervous system only. Okay. So any doubts here? I know it's been pretty long. Any doubts here? Is it clear? Any questions? All right, good. I hope we are almost there. So this is what structure and functions of nervous system. Okay. Do we have another one? I think we have to finish this and go. Anyway, so we'll quickly finish this up. So this is like very simple. This is about structure of the nervous system. Most of you may know it. So the anterior part is mostly for movement, and the lateral part is mostly for perception, more automatic functions. So anterior part is more executive and output oriented, and more the posterior you go, you have more automated perception and memory based functions. Okay. So anterior part, which is predominantly your uh, frontal cortex, prefrontal cortex, and frontal lobe, is involved in movement. Corpus callosum is a part of telencephalon, which is the cortex. It connects cortical structures, as in left and right hemisphere. Diencephalon is basically your brainstem's top, hypothalamus and thalamus. Mesencephalon is the midbrain. Metencephalon is the hindbrain. Mile is the brainstem. So metencephalon includes your cerebellum, pons, those things. So right answer here would be telencephalon. if we do need a picture, yeah, this would definitely need a structure. Just a second. So here you have it. So the spinal cord and just this medulla, it would be your mild encephalon. Your pons and cerebellum would be your met encephalon. This blue structure here would be your mesencephalon. So the top of the brain, which has the thalamus and hypothalamus, would be your uh, diencephalon. The rest of the covering, the cortex and the connecting structures are all telencephalon. Okay. So corpus callosum, which is a cerebral commissure, which is connective tissue, is a part of telencephalon. All right, hope that clarifies a lot of things. All right. So subcortical structures, which are involved in motivation and emotion, are basically your limbic system. All right. So these are all still subcortical in a way, but they are part of the mesencephalon. Basal ganglia is for movement and learning of movement. Limbic system, which involves the what do you call it, hippocampus and amygdala, they are for motivation and emotions. Okay, moving on. Fissures are basically large sulcus. Sulcus are drops and gyri are bulges. So fissures are large sulcus. Which fissure separates primary motor cortex and primary somatosensory cortex? You have the central fissure. Another word for it is Rolandic fissure. I don't have the name for Rolandic fissure, but still, yeah. If we can have this picture. So this distinction between the blue and the pink regions, so that is a central fissure. The first strip of the cortical strip, you could say, behind the central fissure is your somatosensory cortex. The one before it on the right side is your primary motor cortex. They're kind of adjacent to each other so that they can coordinate better. So the central fissure or the Rolandic fissure separates them. So similarly, you have the lateral fissure here that separates temporal and your frontal brain. Okay, so these are the major divisions. Which of the following is responsible for coordinating movement in relation to moving objects? So it is not primary motor cortex because it does kind of coordinate before producing a movement, but after a movement is produced and you have to move it, adjust it for a moving object, it's predominantly the cerebellum. So cerebellum is also involved in balance. It's also involved in coordinating movement, maybe also motor response learning, and also the basal ganglia is involved in it. Right. So which of these cortical areas are likely to be damaged by or least likely to be damaged by brain injury? So it's not primary visual cortex because it's just right at the back. Somatosensory cortex is also exposed. Motor cortex is just adjacent to it. The primary auditory cortex, which is also called Heschel's gyrus, is kind of well hidden within it. So temporal lobe, if you look at it, so here is a temporal lobe. There's more structures inside it. So it is not easily see. So this is the Heschel's gyrus, is mostly hidden from view. So primary auditory cortex is more safer on the inside. It's least likely to be, affected by any concussions or any whiplashes or even any damages to the brain or traumatic brain injury and things like that. So that is least likely to be damaged by brain injury. All right. So then we have areas of the brain responsible for taste perception. Of course, gustatory. So it is insular cortex and the base of primary somatosensory cortex. So the base of primary somatosensory cortex kind of goes inside the temporal fold. So it is insular cortex and the base of primary somatosensory cortex. All right. So dash is the part of the brain through which all sensory nerves pass. So thalamus, which is the iliac center of the brain, through which all sensory nerves pass and then go to the respective cortical areas, except for the olfactory nerve. or olfactory cortex, which starts at piriform cortex and then goes to the thalamus. All right, so that is one exception. So dorsal part of dash is called dash, while the ventral part is called dash. Okay, there's so many dashes. I should not have put it. Even I'm confused right now. Dorsal part of dash is called dash. Okay, we have tectum, tegmentum. All right. So tectum, tegmentum are basically midbrain structure. The first dash would be mesencephalon. So dorsal part is the top part, and the ventral part is the bottom part. On top we have tectum, and then bottom we have tegmentum. So we can find it in the midbrain structure. Okay, so this is the midbrain structure. So ventrally, you have all these structures, and at the top, you have the tectum. So tectum is the dorsal portion, is at the top. It has your superior and inferior colliculus. And tectum, which is at the bottom, has all these rest of it, substantia nigra, you have red nucleus, reticular formation, and even the aqueduct. Right. So all these are part of the tegmentum. So that is one thing to remember. So dorsalis tectum, ventralis tegmentum, and they are part of the mesencephalon, which is midbrain, not met. Met would be part of the hindbrain, so it is not that. Nerves emerging from the ventral portion of the spinal cord. So the spinal cord has the ventral and dorsal portions. All right. The ventral and dorsal are for motor and sensory functions. I think ventral is more ventral ganglion, right? So even I'm confused. I need help here. I feel it's sensory functions for ventral and dorsal is motor. You just check it. And I'm not completely sure of this, but I just want to check it. We have the spinal cord. Okay, we have the ventral and dorsal sides, but what goes where? So we have the ventral root and the dorsal root. So all dorsal are sensory. Okay, so it was the other way around. This is a bit confusing for me also, for the love of God. I cannot get it right because it's just two things, right? So maybe you can make a meme out of it and everyone can understand how it goes. So the ventral part is motor. So it has no ganglion. The dorsal part is for sensory. So all the things are collected here in a ganglion, or you could say cell bodies, and then goes to the spinal cord. So this is something even I had difficulty with. All right. So that would be motor for ventral and sensory for dorsal. All right. So parasympathetic neurons, they have a longer first stage and a shorter second stage neuron. Okay. So reason is that they require quick actions on putting brakes. So they have a shorter second stage neuron. So stage basically means the tracts or the nerves, they end up in a set of cell bodies, which is ganglion. And from there, they go to the spinal cord. No, not spinal cord. They target regions like adrenal cortex or pancreas or so many other target regions. The second stage are way more shorter so that they can apply brakes much faster. So that is one reason for that. So ganglion root can be found on the dorsal side of the spinal cord, which we just saw. And it is mostly made up of multipolar neurons. Now, the cerebellum, which is called like a little brain, so it is connected to pons. Pons is the other side opposite of cerebellum by a set of strips of axons, you could say. And they are three. So they are called peduncles because they look like little feet. I could never find that in the pictures, but they're called peduncles. So there are three pairs of peduncles. All right. We have a picture of it. Yeah, we do. All right. Okay, so we don't have a picture of it. So sorry for just switching. So they are attached to peduncles. Yeah, there are three: superior, middle, and inferior peduncles. So, but we don't have a picture of it, unfortunately. Oh yeah, we do here. So you can see superior, middle, and inferior. kind of, you can see that. So they connect cerebellum to the pons. All right. So damage to cerebellum results in jerky, poorly coordinated, exaggerated movements. It can also happen because of basal ganglia damage, but there are differences between them. In cerebellum, you will have initiating tremors. When you move towards an object, you will have tremors. And in basal ganglia damage, even when resting, the body is not able to maintain adequate tone. So even when resting, they'll have tremors. So that's a major difference between basal ganglia damage, which is Parkinson's, and cerebellar damage, where more intention tremors are there. So that is one. And they may also have difficulty looking up and down. And nystagmus can be present because of that. Nystagmus is eye movement. So these are all some cerebellar signs. Okay. The structure that is present in both basal ganglia and limbic system is amygdala. So yeah, easier. Okay. So now we talked about basal ganglia, right? So disorder in which the pathway from substantia nigra to the striatum in basal ganglia is deteriorated, resulting in rigidity, tremor, and poverty of voluntary movement is Parkinson's. They also have tremors, basically resting tremors most of the time. It's because of Parkinson's or low levels of dopamine in them. So reticular formation is not present in which division of the human brain? So reticular formation is a structure that is involved in arousal. When we talked about sleep, and raphe nuclei and all are actually part of the reticular formation. So they involve in arousal and they're part of the brainstem, they're part of the cerebellum and pons, and they're also part of the midbrain because you did find the reticular formation in tegmentum, right? But they are not part of the top of the brains or the top of the brainstem, which involves thalamus and all that. So it is not present in diencephalon. Lastly, somatosensory cortex is present on the post-central gyrus. Pre-central gyrus is though the motor cortex. So this is about the brain structure. Are we done? Oh my God, we do have more. Fine. We'll push through this. Any doubts or questions? We'll try to rush through this
What
so that
was
we can.
the answer of the last MCQ?
Sorry,
What was the answer of the last MCQ?
last one, previous. We'll go back.
Yeah.
Somatosensory cortex is on the post-central. Okay. Thank you for pointing out. When I was explaining, I just clicked pre-central. So it is on the post-central gyrus, right? Just behind the Rolandic fissure or the central fissure. Then you have the gyrus. So that's the post-central gyrus. Pre-central is basically your motor cortex. When I was trying to explain, switched. So it is post-central gyrus. Okay. Pre-central is motor cortex. Anything else? Is it clear so
No,
far?
thank you. Yes.
Good. So now we move to glandular system. We'll try to rush through this. We'll not go back and forth to the PPT or something. So glands release their chemicals, which are called dash directly into the circulatory system, which are called dash. So directly into the circulatory system are called endocrine. Okay, so when you release things directly into the circulatory system is endocrine, but if it is released through tubules, it's called exocrine. So they release hormones. Okay, so it's not exocrine, it's endocrine. Hormones are synthesized from cholesterol and they are called steroid. So steroidal hormones are synthesized from cholesterol. So you can find it by steroids. So that can be a hint also, which is the master gland. Pituitary gland is the master gland, but the pituitary is in fact controlled by the hypothalamus too. So it may not actually be the master gland, but we do have pituitary as the master gland. Tropic hormones are released by anterior pituitary. So the pituitary is two, anterior and posterior. The posterior pituitary releases vasopressin and oxytocin, which have other functions. The anterior pituitary involves tropic hormones, which go and trigger a particular hormone in the target site. Okay. So that is how it acts. So tropic hormones are triggered in the anterior pituitary. So now when we talk particularly about adrenal hormones, what is the correct order? So we'll have releasing hormones and also tropic hormones and the adrenal hormone. So the adrenal hormone is cortisol. So it should be at the end. So cortisol should be at the end. Then we have releasing hormones. And before that, we have tropic hormones. So tropic hormones trigger releasing hormones. And then we have the actual hormone. Right. So you can, fine, we'll just move forward. Which of the following is the correct order of sex hormones? So again, we'll have tropic, releasing, and the sex hormones. So testosterone and estrogen are the sex hormone, which are end products. Then we have releasing hormones from them, and we have tropic hormones. I just have to clarify this to show you. Just hold on for this. So here's the picture of anterior pituitary and posterior. So anterior is for releasing on tropic hormones. Where is that table? Sorry, I think I have to add one more slide for this. Anyway, so when we have the classes for this, we'll add that slide. Sorry about wasting this time. Anyway, so tropic hormones lead to releasing hormones, lead to actual end product hormones. So posterior pituitary, we already saw oxytocin and vasopressin. They're both from the posterior. Anterior is for the tropic hormones. Hormones that are also neurotransmitters, dash and dash, and they are dash neurotransmitters. So epinephrine and norepinephrine are also considered hormones. Some people even say dopamine is, but it's not a hormone, but it's mostly a neurotransmitter. But anyway, epinephrine and norepinephrine are neurotransmitters. So they're not amino acids, they're monoamines. Which of the following hormones is triggered by the level of glucose? That will be insulin. Hypothalamus controls posterior pituitary, through dash and dash. Hypothalamus also controls anterior pituitary through dash and dash. So it has releasing and inhibiting hormones. All right, so that is where I had the confusion, which gives inhibitory and releasing hormones. Just a second, guys. Okay, so releasing hormones are first, and then we have tropic hormones. So that is where I kind of had a confusion. I was feeling like maybe something is wrong there. So yeah, we go back to these questions, kind of apply motivated for getting here. So the correct order is we have releasing hormones first, and then we have tropic hormones, and then cortisol. This is the usual way. So similarly, gonadotropin releasing hormone, and then we have gonadotropic hormone, and then the end products. Okay, so this is the correct answer. I felt somewhat of a cognitive dissonance when answering this question, but anyway, so I couldn't find it exactly there. So right now, please understand it's releasing hormone and then tropic, and then you have your end products. So the releasing hormones are released in the hypothalamus. Okay, so releasing and inhibiting are present in the hypothalamus. And hypothalamus controls posterior pituitary through parvaventricular nuclei. And supraoptic, not superior olives. Superior olives are like way below. So two major areas of, okay, so we move into motor system. I think this is the last. Okay, we do have more, right? Anyway, we'll push through. We'll try to take 10 minutes. So two major areas of sensory motor association cortex are supplementary motor and premotor. posterior parietal and premotor. Okay. So it is posterior parietal and premotor. Supplementary is a supplementary motor cortex. It's not an association area. Okay. So these two. And after that, you have supplementary motor cortexes. So mirror neurons are present in the premotor area, the ventral part of it. So it's in the ventral premotor area. So mirror neurons are basically which helps you learn information or copy information just by observing another person. So that is the activity of mirror neurons. And they are part of the premotor area. The posterior parietal cortex does not receive information from olfactory system. It receives auditory information, somatosensory, as well as visual system, but not from the olfactory system because it does not require smell to give a output, motor output. Okay. So dash initiates movement and dash coordinates information after the initial movement to adjust it. So primary motor cortex initiates movement through coordination with primary association cortexes and supplementary motor cortex. And the cerebellum kind of coordinates it afterwards. Right? A type of motor learning that is usually acquired gradually, trial by trial, is called habit learning. So it's not habituation, habit learning. Then dash pathway controls the distal limbs on the contralateral side is how it is controlled. So it's the dorsolateral motor pathway. So there are two motor pathways, just like how we had two pathways for somatosensory cortex. We also have two motor pathways that are descending. If we have time later, maybe you can study that. But for now, this is the correct answer. It is contralaterally controlling and is a dorsolateral motor pathway which controls the distal limbs, the ones that are more away from your body. All right. So bed cells are actually some sort of a pyramidal cell that are present in the primary motor cortex. This one I'm even confused. Let's just clarify this. PET cells in the motor cortex. We have the primary motor cortex. Do we see bed cells here or the secondary motor cortex? We don't see it in neither of them. All right. Anyway, these are the descending pathways which we talked about. So we have the dorsolateral and we also have the ventromedial, which are again divided into two. Okay. Oh, bed cells are also mentioned here, which are part of the primary motor cortex, which is true, correct. So they descend in the ventromedial region of the spinal cord. Control of voluntary movement. Okay. So the ventromedial tracts project to the proximal muscles of the trunk and the dorsolateral project to distal muscles. So we are also correct there. I just had another bit of cognitive dissonance there, but anyway. So Beth cells are also present, are actually present in the primary motor cortex. They are pyramidal. And for the previous question, it is dorsomodal pathway and they control contralateral sides of the body. Right? So dash in a neuromuscular junction is called motor end plate. It's the postsynaptic membrane. It's a motor end plate. Dash acts as cofactor that permits myofibrils to extract energy from ATP. It's calcium, not just in exocytosis. Calcium is also involved in like making ATP available as an energy source. Okay. Yeah, we have the last question. Dash respond to stretch and dash respond to length of muscles. So Golgi tendon, which is the tendons are basically the structures that attach muscles to bones. So they are sensitive to stretch. So Golgi tendon organs are sensitive to stretch. And it's the intrafusion, so which are inside the muscles, they are responsible for the length of the muscles. So if the muscle is lengthened or shortened, they respond to it and give feedback. So that is present for length of muscles, right? So understand this was a bit quick. And if you had any doubts, let me know. Otherwise, we'll just go to the last part of it. Any doubts or any questions? Is it clear so far?
I didn't understand the habit learning MCQ. I thought it
Okay.
would be trial and error learning.
All right. We'll go there. So this is about biopsychology, all right? So it's not about, let's say, learning and memory. So it's a type of motor learning. Okay, so it's acquired gradually. So it's, of course, habit. So something that is gradually obtained is a habit. So trial and error learning is also just for one response. But if you look at it, trial and error is something that happens almost like once. So once you have learned through trial and error, for example, the uh, cat lever pressing by Thorndike. Once the cats have identified lever pressing, they always press the lever to escape, right? So trial and error learning doesn't happen gradually. It's almost immediate. Okay. So you could say habituation and desensitization, but they are not for motor responses, but rather for sensations. So habit learning is the correct answer here. What was the other question?
Yeah, I understood everything. Just this was something that I couldn't. Thank you.
Good. So I thought you had another question also. Anyway, so we'll come to the last part. So hunger and thirst. So, okay. So there's an assertion reason question, which you will see mostly in net only, not in most of MPhil questions. Maybe in IMHANS, you can see, not N, but IMHANS, IMHANS. So satiety mechanisms monitor the activity of system variables. System variables accurately indicate deprivation of bodily requirements. So there's only R is correct here because satiety mechanisms do not monitor system variables directly. They monitor the uh, correlates of system variables. But suppose if you're thirsty, you will feel like your tongue is parched. All right. So that is what leads you to take more water. But parched tongue immediately does not get quenched. I mean, it immediately gets quenched. Sorry, what am I saying? But the blood levels of water, which is the actual system variable, does not change immediately. Okay. So the parched tongue is what is being or triggering the consumption of water here. But it does not exactly or immediately correlate in real time with your blood's water level. Okay. So it is not the exact system variables that are being monitored, but it is the parched tongue that triggers it. That is what is monitored. So only R is correct here. Okay. So match the following about thirst mechanisms. So we have adipsia, lack of drinking, lack of thirst, osmometric thirst, volumetric thirst. So salt appetite usually leads to a lot of volumetric thirst. Sorry,